So let's look at 5:18 now. This is the one I butchered in class. So I'm going to try and redeem myself a little bit right here with this little video. So, okay, so we're looking at Drosophila, and we're looking at two mutations, stubble. And one of the confusing parts of this question is that stubble is a dominant mutation, so we're going to symbolize that SB. Now, SB, unfortunately for Drosophila, is lethal in the homozygous condition, so that is lethal. Now, this genotype, SB, SB+, so SB+ is the wild type allele, but it is recessive to the mutant allele. So these flies have the stubble phenotype. And then we have our normal, let's call this long hair phenotype here, which is the wild type phenotype. And the other mutation we're looking at, or phenotype, is wings, curled wings, and that is simple. So just the mutant allele, two mutant allele CUs here, well, they have the curled phenotype. And I'm guessing this is the wings, so let's assume it is the wings. So CU+, CU, normal wings, normies. Call those flies normies. My daughters watch a show called, I think, Monster High. We have the monsters versus the normies. Okay, so two wild type curled alleles are normal. Okay, so the genotype of the female in question here is SBCU. These are linked together on one chromosome, and the wild type versions are linked together on the other chromosome. And because the book uses just the pluses, I'm not going to put SB and CU here. We'll just use pluses, and if the pluses are underneath the SB, well, then we know that's a SB+, and a plus under here is really the wild type CU+ allele. Okay, so the question is, if this female with this genotype, was going to be mated to detect recombinants, we want to map. We want to see what the distance is between these two genes, and we want to cross this to a male, with a certain genotype, and we want to be able to detect recombination between these two. What genotype are we going to pick for the male? So, I mean, there are a number of possibilities, right? So we could use SB CU over SBCU, right? Can we use that one? No, we can't, because this is lethal. How about SBCU over plus CU? We could try that, but I think one of the things that is probably going to cause that not to work is that, well, these two gametes are different, and they're going to give us a different phenotype in the progeny depending on the egg they fertilize. I mean, if SBCU fertilizes a plus CU gamete from the female, well, the progeny is going to be stubble and curly. But if the plus CU fertilizes that plus CU egg, well, the phenotype is going to be long-haired. That's a wild type for the hairs, and curly. So we're going to have two different results there. So we're probably not going to want that. So I'm going to guess that the genotype we want is going to be plus CU in the male, homozygous for both the wild type stubble allele and the mutant curly allele. it's almost always in these problems we're going to want to use the homozygous recessive alleles for both genes. But in this case, because stubble is dominant, we are looking at a little different of a situation here. So the wild type is recessive. So actually, when you look at it, we are using the recessive alleles, homozygous recessive alleles for each gene. So let's make sure this works. So this female here can make how many types of gametes? Let's see, 4 types of gametes. So SBCU is a non-crossover. ++ is a non-crossover. And then we can get crossing over gametes too. We can get SB+ and +CU. Now the most important thing about this male genotype here is it has to allow us to determine the genotypes of all of these gametes by looking at the phenotypes of the offspring, right? So when each of these gametes is fertilized, we have to be able to tell the genotype of these gametes by looking at the phenotypes of the progeny. So let's see, the progeny over here have to have, in this case, when this is fertilized, it has to be stubble and curly. Because when we see that, we're going to know that that egg had the stubble allele, the mutant allele, and it had the curly allele. These ones right here, when this egg is fertilized, the progeny have to have a wild type phenotype, because that will allow us to know that the female gamete here, the egg, had a wild type stubble allele and a wild type curly allele. For this one right here, the progeny have to have the stubble phenotype, because then we'll know that this egg had the stubble allele and the wild type curly allele. And for this one right here, the progeny have to have the curly phenotype, because that will allow us to tell that this egg had the wild type stubble allele and the mutant curly allele. So just by counting up the numbers of flies with these different phenotypes, we will know how many of the eggs had these particular genotypes. But we have to choose a male that has a genotype, so that when we fertilize these eggs, that these phenotypes come through. So we chose a +CU male. So the +CU male only has one type of gamete. So we need to just combine the plus stubble allele and the mutant curly allele with each of these over here and make sure we get stubble and curly. Or make sure we get these progeny phenotypes in each case. And you can see that we will do. We'll get that. So when we're looking for stubble and curly, we can see this SB is dominant to that plus, so these progeny will be stubble, and this CU and this CU are both recessive, so they will be curly. Okay, perfect. So every time this egg is fertilized with +CU, the progeny are going to be stubble and curly. And great, we're going to be able to know the genotype of that egg in each case. For this one, when a plus and a CU fertilizes an egg that has the wild type stubble allele and the wild type curly allele, well then the progeny are going to be wild type, because we don't have the dominant stubble allele and we don't have homozygous recessive condition for the curly allele. So let's take a look down here. When this +CU gamete fertilizes eggs with a genotype with the SB mutant allele and the wild type curly allele, well, SB is dominant to SB+, so we see the stubble phenotype, and we don't see the curly phenotype because plus is dominant to CU. Okay, so this one checks out. And you'll see that the same thing occurs for this one. So we want to choose this genotype for the male because it allows us to just look at the progeny phenotypes and be able to infer the genotypes of the female gametes.